Richard Barker, as Monty Python might have said, now for something completely different. After George Church's actual real tour de force. George, thank you again for that. So this is a foundational session on the hypothesis that molecular and cellular damage and the repair of damage lies at the heart of the aging phenomenon, and that there are linkages there. And I have at least one advocate of that position sitting very close to me here. By the way, my name is Richard Barker, and I head an institute between Oxford and UCL on a different central mystery, which is why, given all of the fantastic technology we've seen over the last 30, 40 years, we still have precious few really transformational therapies coming out the other end. I think George hinted at some therapies that might well come out the other end. But so we work on what I call medical innovation science. What's the science of this whole process? Since I've been observing this field for about 30 years, when biotech was recombinant DNA, which seemed to get out there quite quickly. So what are we going to do to transform the process by which fundamental breakthroughs get through to human patient benefit? So that's what I do. The gentleman to my left probably needs no introduction. The absolute pioneer in this field, Aubrey de Grey, who will go first with his contribution. Then Jeff Karp, who is a man of many parts, difficult to pin down, works, as you will have seen in your bio, with Harvard Medical School and Brigham and Women's in a whole range of areas in terms of devices, nanotechnology, drugs and drug delivery, a whole range of things. He'll no doubt talk about that in his brief intervention. Stephen Minger, who has a very distinguished career in stem cell development, some of it academic with King's College London, but is now with GE, and having pioneered the stem cell area in GE, he is now the Chief Scientific Officer for the life sciences field. And then last, we'll go Caleb, who is, as Aubrey put it, the card-carrying gerontologist on this group. But again, as you will have seen, he also has appointments in anthropology, molecular biology, neurobiology, psychology, physiology, and neuroscience. Have I got them mainly? So when we get to the question and answer thing, he will not be surprised if the majority of the questions get fired in his direction. So the format will be each of my distinguished colleagues will make a brief presentation. I think only Caleb has slides, and that's why he's going last. And then we will have the free-for-all that we had, hopefully with the same level of stimulation that we had after George Church's excellent talk. So with no further ado, Aubrey.
All right. So yes, I'm not going to give slides, but I've just written myself some notes to kind of respond, actually, in some ways to some of the things that both Mike and George have already said today. Actually, one of the reasons I have decided not to use any slides is to emphasize the fact that we've got quite a lot of these discussion panels in the agenda for this conference. We've very much decided that it's going to be useful in an extremely multidisciplinary conference with an extremely multidisciplinary audience and set of delegates to promote cross-fertilization. So there's, just as in this panel we have 2 gerontologists and 2 not really gerontologists, we're going to have a great deal of exchange between areas of expertise. The slides that I would have used if I had chosen to would be the same ones that you can find on YouTube in a million places anyway. But I'm just going to start by reminding you all of something that actually was the first source of frustration that I had when I got into gerontology. And it was that even people who study the biology of aging have a wide range of definitions of the word aging in their heads. If you ask them what aging is, you will not get the same answer from everybody. This is frustrating because it actually gets very powerfully in the way of dialogue and debate. And actually, in some cases, I personally believe that experts in the field have a sufficiently confused idea of what they think aging is that they end up holding views that make no real logical sense. I have a trick that I play very often with when people say things that and I have a feeling they don't really know what they mean. I just tell them to restate what they just said without using the word aging. It's a very clever, it's a very powerful technique, I have found. So my definition of aging is really simple and down to earth and boring, really. Aging is the accumulation of damage to the body as a side effect of being alive in the first place, as a side effect of the body's normal operation. And the reason that's a useful definition is because it is one reason anyway, is because it emphasizes how similar the aging of the human body is to the aging of simple man-made machines like, cars or aeroplanes. Essentially, it's just a law of physics, forget biology, that anything with moving parts is going to damage itself in the course of its normal operation. And of course, eventually this damage exceeds the quantity that the body has been set up to tolerate. I want to parenthetically stop for a moment here and mention something that came up in the Q&A in George's talk, which is the question of whether aging exists for a reason. In other words, whether evolution has taken the trouble to create active processes which eliminate the older members of the population, essentially in the interests of non-individual fitness benefit for the population as a whole. I personally think, and I believe actually most people who study the biology of aging think that that's not the case, that aging exists, as somebody said a long time ago, as a consequence of evolutionary neglect rather than evolutionary intent. But the thing I want to focus on right now is that whether or not aging exists because evolution wants it to, that's not in any way a question, that's not the same question as, does aging get driven by the accumulation of damage? The fact is, whether or not aging is programmed, as some people would put it in one word, it still is driven by the fact that eventually the body gets into a state which is prejudicial to optimal function. There's another thing that I want to emphasize, which relates, I think, to some of George's remarks, and which we'll probably be coming back to quite a lot during the conference, which is, how do you actually figure out what really matters in aging? And one mistake that a lot of people have made over the years is they think that you can find out a lot about aging by studying really old people. That turns out, in my view anyway, not to be the right way to go, because really old people have so much going wrong in their bodies, and those things that are going wrong are interacting with each other and exacerbating each other in such complex ways that unpicking all of this and gaining any kind of understanding from it is pretty hard. Whereas, if we study young adults instead, if we study what is the actual difference between, let's say, a 20-year-old and a 40-year-old, then we have a much better chance. We know that there is a difference because there's got to be some physical reason why a 40-year-old has a considerably shorter remaining lifespan than a 20-year-old. But nothing much has actually arisen from that difference yet, which means that it can be studied in a much clearer way. That's essentially the kind of thinking that led me to this 7-point classification that actually George put up in one of his slides of the types of damage that accumulate throughout life and that eventually drive the ill health of old age. Something that George spent some time on during his remarks was epigenetic change, which is undoubtedly something that's become far easier to study as a result of new technologies such as the ones George mentioned. I want actually, in that context, to mention something that also came up in questions, which is the issue of whether rejuvenating the epigenetic state of tissues in a selective way or an indiscriminate way would be a good thing or not. I have personally come to the conclusion that, by and large, it would be a bad thing, just as the questioner suggested, and that the only epigenetic changes that we should really be concerned about in respect of aging as a contributor to aging are ones that happen adventitiously, ones that you might pick up as epigenetic noise, changes in the epigenetic state from one cell to another that are nominally of the same cell type and should ideally have the same epigenetic state. This is something that actually we've been funding for a few years, the study of at Albert Einstein College of Medicine. But I think, in general, that if something is seen in bulk, if it's seen as a coordinated epigenetic change in a tissue, then it has happened for a reason. It's happened as a result of some kind of genetic response to the cell's environment, whether inside the cell or in the circulation, in the context it finds itself in. And as such, it's probably happened in order to optimize the health of the cell rather than anything contrary. Really, the main thing I want to close with in these introductory remarks is to make a linkage between the damage repair philosophy that Sense Research Foundation follows and the philosophy that the overwhelming majority of the medical community and the medical research community follow when they look at the possibility of doing anything about age-related ill health. Of course, what everybody does, virtually, where virtually all the money goes, is to treating the individual ailments of old age like diseases, as if they were like infections, things where you could bash away at the symptoms and eliminate them from the body in the same way that one might eliminate tuberculosis. We all know that that's inherently crazy, that since these things are side effects of being alive in the first place, you're not going to cure them in that narrow sense of the word cure without curing being alive in the first place, which would rather defeat the object. So really, we've got to look for a different philosophy. And the damage repair philosophy is that. It's preventative in the sense that it's focused on eliminating the precursors of age-related ill health. But something that I don't often highlight, and I think it's perhaps useful to highlight it in this meeting, is that not only can these therapies be used preventatively, when we develop them, on people who are not yet sick, but also for people who are already in perhaps even quite an advanced stage of age-related ill health, it may be possible to potentiate the activity, the efficacy of geriatric medicine, of traditional medicines of the sort that currently don't work. The reason they don't work at the end of the day is largely because as time goes on and as the precursors that drive these diseases and disabilities continue to accumulate, so inevitably, inexorably, any geriatric medicine is going to become progressively less effective. There's just more to combat, to counteract. So if we can solve the underlying precursor problem as well as addressing the pathologies, we may have a great deal of synergy between these therapies. This is something that perhaps exemplifies the combinatorial nature as well as the preventative nature of how I believe aging is likely to be brought eventually under comprehensive medical control. And of course, it is a very big departure from the way that people like to do medicine today. Medicine, medics understand that medicine is black magic, and they try to keep it as simple as possible just in order to have some idea what's going to happen. But at the end of the day, and especially with new technologies of the sort that we've already heard about this morning, I believe that we will be able to start being more courageous in this regard and actually contemplating and working with highly combinatorial divide-and-conquer approaches that really have a respectable chance of bringing aging under proper medical control. So that's all I'm going to say for the moment. Thanks very much.
All right. So when I was asked to speak here, I thought that I would be asked to talk about some of the bioengineered strategies that we were developing in the lab. And then as it got close to the conference, I looked at the title of the session that I was speaking in. It says, Molecular and Cellular Damage as the Cause of the Disease of Aging Panel. And I was literally shocked. And so I started to think, okay, well, how can I potentially connect what we're doing in the lab with this panel? And then I recalled a paper that I had read maybe 4 or 5 years ago. And this paper was written by Alan Spradling, who some of you know is a Howard Hughes investigator. And the paper was kind of outside what we were doing in the lab at the time, but I was just fascinated by it. And essentially, it's Alan Spradling, he studies the Drosophila ovary. And in this model, what happens is that you have stem cells on one side and differentiated, there's progenitors and differentiated cells, but then you have the mirror image on the other side of this niche. And what happens is quite remarkable, is that one of the stem cells on one side of the niche, it divides, and then the stem cell crosses over to the other side. And if a stem cell in the other side is damaged, then it will evict that cell out and take its position. And if that position is vacant, then it can take that position. And if there is no vacancy or no damage, then what happens is it differentiates and fills a spot below that stem cell. So this is an example of long-range stem cell replacement. In fact, that was the part of the title of the paper. And so clearly, I think that this could be relevant to replacing aged or damaged cells, stem cells, using in humans in some sort of cell therapy-based approach. And so, I started thinking a little bit more about it, and I think that we know that as we age, the number of stem cells in our bodies decreases, and we know that there's a progressive loss of stem cell functionality, and I think this contributes to three things. So one is increased degeneration. So tissues are less functional. The second is decreased regeneration. So we're not able to heal damaged tissues as well. And then three is a declining ability to maintain homeostatic functions. So in thinking this through, I think we might be able to connect this decline of function of stem cells in our body or decreased numbers in their existing niches with the ability to potentially populate niches with young, healthy stem cells. So to continue on that thought, we all know that with age, niches become less supportive to maintain a stem cell pool, and that as we age, there's a greater need for stem cells to really work overtime to try to replace our aging tissues, damaged cells. So I'd like to argue that there's an opportunity to engineer cells to specifically deliver them to sites in the body to do four things. So one is to fill vacant niches. Two is to replace damaged stem cells or progenitor cells with new ones. Three, to fix damaged niches. And four, to create new niches. And I think that achieving any one of these could be used in an attempt to get those who are old to essentially regenerate as if they were young, and to potentially bring people back into homeostasis. And I think this isn't such a far-fetched idea for a number of reasons. So there's significant evidence that ex vivo manipulated cells are capable of repopulating distant niches and creating new niches. I'm going to provide some examples. So one, I think the best example is hematopoietic stem cell transfer, or bone marrow transplantation, or peripheral blood stem cell transfer, where you have bone marrow or cord blood, and you can deliver these cells into the systemic circulation, and they will target the bone marrow in a patient that's been irradiated, so they have no capability of forming new blood, and those hematopoietic stem cells will either take on existing niches or create their own niches to repopulate the entire, to basically restart the entire blood-making system. So I think that's a great example of an exogenous population of stem cells that can then form niches at a distant location from the infusion. Hans Klevers, who many of you know, studies intestinal epithelial biology, and he showed in a very interesting model that if you take LGR5 positive stem cells, so these are the cells that are at the base of the crypts in the small intestine and in the colon, and if you take these cells and you expand them, so you're manipulating them outside the body, and then infuse them into a mouse with ulcerative colitis, so an ulcerative colitis mouse model, that these cells can essentially attach to the ulcerated tissue, engraft, and form new crypts that will continuously supply differentiated epithelial cells for at least 25 weeks. And I don't think he went beyond that, but for 25 weeks was able to show that. We also know that there's emerging evidence that mesenchymal stem cells can take on pericyte locations. So MSCs are believed to be derived from pericytes, and there's emerging evidence that when you administer MSCs, that they can then attach to the blood vessel surface. Maybe doesn't do it very efficiently, and potentially that's because we don't have a good handle on on controlling the MSCs, the biology of the MSCs, and how we can maximize their ability to take on pericyte locations, but that's been demonstrated. And then there's also examples of taking satellite cells and infusing them, so manipulating them ex vivo, infusing them and forming new niches in muscle to generate new muscle tissue. And then I think also to kind of fuel this possibility, we know that in cases of cancer metastasis, that likely the case in certain types of cancer that we have cancer stem cells that are migrating to a distant site in the body and forming new niches at those sites. So I think that some of the technologies that we are developing and many others are developing potentially could be useful to try to create new niches or to replace cells that are damaged or aged in existing niches. And in a way, you may think of this as potentially editing niches for therapy. And so just to describe some of the technologies that we've been working on and some of the others that I think might be able to address this. So now we're at the stage where we can actually equip cells outside the body with microparticles or nanoparticles and load small molecules or other agents in there so that after you deliver the cells, you can control the cells regardless of where they are in the body because those particles are associated with the cell and they continuously deliver that factor that you've put in there. So that's a way to control cells following transplantation. There's ways to target cells systemically. So we've developed a number of technologies where we can modify the cell surface and then you do systemic administration. You can target cells to distant sites in the body. So for example, we know the zip code of inflammation. We know that we have E-selectin, P-selectin, ICAM, VCAMs highly expressed. We know in the gut, in the endothelium of the gut, we have MADCAM1 that's highly expressed. In the peripheral lymph nodes, peripheral node addressins. So we know some of these zip codes. We potentially can just target cells to those sites. And then we also have technologies that we've been developing where we can put sensors on the surface of cells to better elucidate the niche biology. So use intravital microscopy and then be able to watch cells communicate with each other in real time at the single cell level in an animal model. So I think there's a number of challenges to address, and just in closing, I'll mention some of the challenges I think to try and do this. So one is to be able to generate sufficient numbers of cells for transplantation. The other is to try to, how do we develop approaches where we can maximize the ability of cells to survive and engraft. Many transplanted cells die following delivery. If we're using an allogeneic source of cells, how do we overcome the immunological barriers? And those actually exist for mesenchymal stem cells as well, despite what many papers in the field say. And then I think another opportunity is to better understand the target sites. So can we better understand niche biology and through understand, or how to create a new niche in a certain site in the body? And I think the more we can better understand these things, the more we may be able to address this concept of having an exogenous population of cells that can replace damaged stem cells in the body or repopulate vacant niches. So that's all I had to say. Thank you.
So like Jeff, I think when I realized what this panel was about, I was sort of thinking, what the hell am I doing on this panel? But then I know Aubrey, and I have known Aubrey for quite a long time, and I thought, okay, so think provocatively. And so I try to think like him. And then I thought, actually, I do relate to this, and I relate to this historically with a lot of the frustration that Aubrey experienced, because I kind of turned my brain back to when I was doing my PhD back in the late 80s, and I did my PhD at Einstein with some really eminent neuroscientists and neuropathologists who were working on trying to understand the etiology of neurodegenerative conditions like Alzheimer's and Parkinson's. And I was really excited about this. You know, particularly in Alzheimer's disease, here's the most important part of your brain, and it's extremely susceptible to this disease that robs you of who you are. And I just thought the whole idea of here we are, these higher cognitive beings with these tremendous brains, and yet, you know, I forget what the actual number is, but let's say 50% of us, if we live to be 85 or not, will dement. And that's a huge number. And so how do we have these complex brains and yet they're so susceptible to disease? So I was really excited about the chance to try to understand what causes diseases like Alzheimer's disease and understand the biology and the biochemistry. But in the late 1980s, there were no animal models of Alzheimer's disease except for surgical models. This is before the era of transgenic mice. So we had no disease models except, you know, going in and ablating specific regions of the brain. Well, that's not very useful. That has no analogy really to the disease itself. The only other alternative was to study people with the disease, but you can't get access to their brains. You can only get the end result, which is a person who maybe died, you know, terminally demented and in a nursing home for 5 years, and you look at their brain, you know, that's been ravaged by a 20-year disease, and what does it tell you? It tells you nothing about mechanisms. It tells you nothing about disease progression. And so I was getting increasingly frustrated because, like, how am I ever going to understand this awful disease and what causes it with the tools that we have available? And moreover, I worked for a very iconoclastic neuroscientist named Peter Davies, and so we were neither Taoists nor Baptists, and everybody had divided up into camps whether amyloid was the most important part of the disease or phosphorylated tau that causes neuronal destruction. Because Peter said, you know, by the time somebody begins dementia, it's all over. What happens occurs 20 years before that. So, you know, I thought, geez, how am I ever going to find anything out about this? So I was very frustrated. And the change was that all of a sudden groups in Sweden, together with a bit of work in the US, began to show that even without understanding Parkinson's disease, you could get tremendous clinical improvement in patients by transplanting human fetal tissue into these patients. And so over a series of about 10 years, maybe 30 or 40 patients in Lund were transplanted, some of which now, 25 years later, still have surviving functional transplants. These patients still have ongoing Parkinson's disease, but their transplants worked and continue to work such that in patients who got sufficient amounts of fetal tissue, they've never gone back on levodopa and anti-Parkinsonian medication. And then I thought, well, here it is. I don't need to understand how Alzheimer's disease and other age-related disorders mechanistically happen. What's important is, can I do something about it? So we moved into, I went to Rusty Gage's lab and you know, gene therapy and transplantation approaches. We pursued that all through the 90s. The advent of human embryonic stem cells, I think, fundamentally changed the field because all of a sudden now, rather relying on fetal tissue and trying to figure out how you take adult cells and turn them into a variety of cell types, you now have a source population of cells that you can grow to unlimited numbers. And you can transplant, you can differentiate those cells, at least in principle, to almost any cell type in the body, and then use those cells therapeutically. You know, people have their own feelings about human embryonic stem cells. Certainly in the UK, we had regulations that were very strict, but very scientifically permissive to allow for the development of human embryonic stem cells, where here in the US it was very, very difficult. And then, of course, with the advent of iPS cells, you now take the technology of ES and ES differentiation, and you now apply it to, in some cases, disease-specific stem cells. So I think we now have the tools to begin to understand some of the questions that Aubrey and others are really interested in, where you now have tools in a dish where you can begin to understand disease pathogenesis and develop therapies around that. To kind of hearken to what Jeff was just saying, you know, cell therapy and regenerative strategies are here. If you go to clinicaltrials.gov and type in stem cells, there are over 4,000 listed clinical trials now, and these are mostly just American trials. Foreign researchers are not obligated to enter their details into this database. You have ACT claiming to get reversal of age-related macular degeneration, which is a progressive disorder. Again, large numbers of people, if they live long enough, will go completely blind, and there are very limited treatment options currently available. And we can now make retinal pigmented epithelium from ES cells or from iPS cells, and people are claiming to get reversal. There are trials in spinal cord injury, there are trials in motor neuron disease, there are a number of ES-related and iPS-related trials going to the clinic. Evidence that this field is becoming progressive and important is the fact that General Electric, of which I'm a chief scientist for life sciences, is a cell therapy company. So think about that for a moment. What does GE do? We make light bulbs, we make dishwashers, we make jet engines. We are now a cell therapy company. We're engaged with huge numbers of clinicians who are in the clinic, both in terms of cancer and in other regenerative conditions. One of the things that I'm most interested in, and I'm driving a program in our own organization, is the idea that you have stem cells in the adult human brain, which every day give rise to new neurons. And there's at least some consideration that these cells are responsible for the formation of new memories. 20 years ago, that would be hugely heretical. The idea that you make new neurons every day, but we know it's true. But we understand nothing about this process except in rodents, and we're not rodents. So what happens in the human brain is completely different. But where are the tools, again, that allow you to monitor what's going on in the human brain in terms of the generation of new cells? We know this pathway gets activated massively in clinical conditions like stroke, where you see literally millions of cells within the niche, as Jeff referred to, where we have stem cells in the adult human brain, which migrate selectively into a region of damage following ischemic insult. But what happens with those cells when they get there, we have no idea. We have no capability to monitor, track these cells. So one of the things we're trying to do within GE Healthcare is use our fantastic in vivo diagnostic imaging around MR and PET to be able to follow these cells, use imaging technology to see where they go and what they do, and can we manipulate this system? Instead of patients with stroke, you give them a small molecule that mimics what stroke seems to do, which is to activate this system. And maybe in cognitive impairment situations, in Alzheimer's disease and others, once our diagnostics get better and we can predict which patients may develop Alzheimer's disease, you start giving them stem cell drugs to activate these cells and hopefully counterbalance the loss of neurons that are occurring in the disease. So I think there are a lot of strategies to attack age-related disorders and aging in and of itself as well. So with that, I'll stop and look forward to questions. Thanks.
So I'd like to talk about this larger topic in an environmental sense, but I would like to first, am I pointing this in the right direction? I'm not getting a response to the next slide. Oh. So there are three points that I'd like to bring up, and I have some images to support this. Here we are working very hard to increase life expectancy, and in some favored populations it's approaching 90. But, and this is a huge point, by 90, the majority of people have clinical cognitive impairment. So as we want to increase to a goal of a life expectancy of 100, we have to confront the issue of the fact, and it's very solid, that the rate of dementia after the age of 60 increases faster than the rate of mortality. So the mortality doubles every 7 to 8 years. Dementia doubles every 5 years. So it's appropriate that in this meeting, 2 high-level sessions are devoted to Alzheimer's disease, because we are heading for a very unpleasant collision with cognitive impairments as we expand the pool of people who are healthy enough to reach 90 or 100. Second point that I want to make is that in the next 50 years, where our environment globally is going to deteriorate on 2 vectors. One is the immense ecological consequences of the temperature shifts called global warming. The second is that increased use of fossil fuels is going to double. And that increases environmental pollution. And I will very briefly in a moment take you through a new front of environmental science that I'm involved in which pollution particles from combustion engines interact with brain development and brain aging, and the evidence looks as though it accelerates amyloid deposits and cognitive decline. And third, something that I don't think it has been given the attention that it merits, and that's we have a likely expansion in the gap of health spans on socioeconomic basis. The technological marvels of regenerative medicine are not going to be as cheap as an aspirin. And I think that there is a huge polarization that will be on every country's political high table about the access to elite medicine. Those of us in the room have that access, and we have far superior health care than the people who are tending the floor in this hospital. So this is a political bombshell that's ticking away, and I think that it's, we need to be farsighted about the political implications of the elite medicine that we're in the process of designing. So let me just give you a little illustration of the Alzheimer's. Actually, heart diseases have been declining for the last 40 years. It's about 2 or 3% per year, but Alzheimer's is climbing, as you can see, and that is largely a consequence of increased survival to later years. This is all in a 40-year time frame. So if we take a global view of where our life expectancies were in 1800, it was about 40. With all the improvements of sanitation, nutrition, and medicine, we've added a year per every 4 years of historical time. And some of my colleagues are positively euphoric that this is going to continue into the 21st century. And I am among a small group who are saying, besides obesity, we have environmental deterioration that is going to affect us and differentially. So here is a summary of a body of literature that air pollution accelerates aging of the arteries and aging of the brain. And there's, I think, besides the particles that are produced by fossil fuel, and I'll tell you a little bit about them, ozone also has this same direction of effect in promoting inflammatory changes and amyloid deposits. So I think we have to confront that we're dealing with a synergy of environmental inflammations with the endogenous inflammatory processes of aging. And that is something that needs to be attacked not on a cellular basis, but on a systemic basis, and the strategies for that may be quite different than the wonderful targeted genetic engineering that we have at hand. So just a glimpse of this. Here is a set of contours that could be drawn for Los Angeles, that could be drawn for any city in the world, that there are zones of higher and lower particle density. In this case, it's Los Angeles, and it's derived from the high level of urban traffic. And very well documented longitudinal studies show that carotid thickening increases 4% for every additional 10 micrograms per cubic meter of particles that are 2.5 microns. The same kind of a contour is emerging for cognitive changes with aging, and I won't go through that, but there is a body of literature now that's at hand on the epidemiology of cognition and aging, that there's 8 papers that document that in higher zones of air pollution particle density, cognitive aging is 2 to 4 years faster. And in the city of Mexico City, comparing a low pollution city with Mexico City, that there is a premature deposits of amyloid in the brains of adolescents and young adults, and of course it follows an ApoE3, E4 vulnerability. So we have animal models for this. We collect air from Los Angeles, store it in sterile suspension, re-administer it to cells in culture, or re-aerosolize it for animal models, and we have quite a complex story now of it showing increased levels in the brain of A beta according to the ApoE levels, ApoE alleles, and also regulation of glutamate receptors, which are keys in cognition. So there's no question that there is a front in Alzheimer's disease, which has now got an environmental context. So looking ahead, the anti-longevity issues that create increased inflammation, obesity, air pollution and inflammagens, dust from deforestation, and I'd like to point out that with global warming, there is an increased need for air conditioning, which is of course driven by fossil fuels. And here's just a glimpse up to 2030 of the air pollution and energy, the world energy consumption. In 25 years, it's going to increase by more than 50%. So the point that I think I'd like to raise for discussion is that we need to think of the environmental context, the ecological context that our efforts to increase health span are going to take place in. And I think that there's another set of multidimensional issues that have barely been discussed. Perhaps that will happen here. Thank you.
So I'm going to throw a few questions around the panel and then open things up. I mean, Aubrey, you made an interesting comment earlier. I think it was probably provoked by George's one, which is, you know, is aging a programmed process, an adventitious process, or is it actually a beneficial adaptation to the environments that the cells encounter as time goes on? And you made the assertion that doesn't matter, actually, right?
More or less.
Do people agree with that?
Well, actually, perhaps I could elaborate just briefly on what I said. It does actually matter a great deal in one way. If there is an active program of gene expression late in life that somehow accelerates the process of functional decline and our risk of death, then we would be very happy. because it's much easier to break something in biology than it is to build it from a medical point of view. Therefore, ideally, if that were true, we would simply be able to intervene in a relatively primitive manner and achieve a substantial postponement of age-related ill health. And that's why, even though I personally think it's pretty much absolutely certain that there is no such program, nevertheless, I give an enormous amount of benefit of the doubt to those who disagree with me. I'll actually give a shout out to Josh Middledorf here, who's one of the most eloquent workers in this area. I believe you got a poster here this time? Okay. Anyway, talk to Josh if you really want to hear the programmed aging concept in detail. However, yes, the sense in which it doesn't matter is the one I spoke earlier about, namely that whether it's programmed or not, it's still a matter of accumulating damage, eventually causing problems. So if we can repair the damage in the manner that SENS Research Foundation is trying to do, then we'll still achieve the appropriate objective.
Okay. Any comments to that? Any disagreement, potentially?
I support this.
Yeah,
That's boring, Caleb, but thank you.
I mean, I think I would as well. I mean, one analogy that I like to use is I have a friend who's a professor at the Chartres in Paris, and he collects cars. And one day, you know, he said, "You want to go for a spin?" And I said, "Yeah." And I heard this horrendous noise, and here comes a 1932 Bentley. Okay, so this car is, you know, pushing 100 years old. And I said, "God, you know, how do you find unleaded petrol for this anymore?" And he said, "This thing runs on anything. It runs on kerosene, it runs on lighter fluid, it runs on barbecue. All it needs is a hydrocarbon chain." And he said, "This car is indestructible. It will last forever because when a part wears out, I just replace it." And I think that's kind of what we're thinking about doing in regenerative medicine. You need some new cartilage, we give you some new cartilage. You get, you know, you need a new hip, we make you a new hip. You know, you get a bit Bidulotti, you know, we give you some new cells, put back in the brain. You just keep replacing the parts. You know, that's not going to keep you from catastrophic disease, but the accumulation and wear and tear of our tissues, I think, can be alleviated if we can make the appropriate tissues and we just replace them as needed.
So your assertion really is that we shouldn't worry about the damage, we just replace the
No,
damage.
no, I think strategies that can prevent that, but it's an alternative if you could, you know, as I said, if you don't understand how it's happening and you don't have tools to impact on it, you replace the damaged bits.
So you two guys work a lot on this hypothesis, which is, can stem cells be placed? And you laid out the 4 environments in which they might be placed. Where are the big scientific challenges that are still remaining in terms of getting those cells to the right place, having them behave in the right way, having them trigger the right microenvironment, all of that? Are we cracking those problems?
I think, you know, on one hand, we don't know enough about where we need to get the cells and what the biology is in that environment. So I think, you know, in some ways, we need better ways of figuring out how we can target niches and then image them and study the biology. And once we have better understanding of that, and I think that, you know, a parallel is also in cancer metastasis. So we still have no clue about how cancer cells can seed other environments and the biology of creating a new niche and then sustaining itself. Like, you know, there's a lot more to learn about that. So I think, you know, there's a parallel in the regenerative, regenerative medicine, where I think we need to better understand just niche biology, and I think that's coming along. But so we have to understand the target site, and then once we do that, then we have to figure out how do we then target to that site.
And there's a big interaction between, you know, the cells that you're putting in the environment they find themselves in. And you can't understand one without the other. You can put the most beautiful cells in the nastiest environment, they're going to get killed. And on the other hand, you know, you can have a really, really rich environment to put cells in, and if they're not the right cells, they're just going to ignore it. So it's a real fundamental interaction that we have to understand.
Steve, I'd like to bring up something I'm sure you are aware of, that in the Parkinson transplants, the transplanted embryonic cells showed the synuclein pathology. In other words, they came in clean, and then they acquired a transmissible protein pathology. So the niche biology of aging is a very different set of questions than the niche biology of young or developing tissues.
Absolutely.
And
That's a very important point.
It interacts
Right.
with these inflammatory processes that are global and tissue-wide.
It's very important.
And I actually have a question related to that, which Jeff reminded me of in his remarks. So the environment that cells find themselves in can be classified into two types. There's the immediate, like paracrine, the niche environment, and there's the circulation, the systemic environment. And I only know of one study that has really tried to distinguish between which of those two has the dominant impact on the health of transplanted stem cells done, young tissue into old or vice versa. It seems, maybe I've just been overlooking stuff.
You mean the parabiosis?
Oh, actually, I'm talking about simply, the study I'm talking about simply took, for example, a fragment of bone, complete with bone marrow, niche, and stem cells, and just stuck it under the kidney capsule of a differently aged animal and determined what happened. And of course, this means that the systemic environment is heterochronic and the niche environment is not. I'm amazed that there isn't more of that kind of stuff going on. Am I just overlooking it?
I think there's a number of studies out there that I think highlight what you've just said. So for example, I think the time of day in which you do a bone marrow transplant actually can affect the engraftment efficiency. So there's some sort of connection with the circadian rhythm, for example. And I think there's these kinds of studies showing that there's some systemic component of engraftment. And so it's not just, I think as you said, it's not just understanding the niche, but then the involvement and the interactions between local and systemic effects.
So let's start to draw in the audience with the following question. Do you mind? We'll get back to you in just a second. So I had a very interesting breakfast discussion with Donald Royal from Texas, who basically asserted that Alzheimer's is not a disease of aging. Donald, are you in the room?
Yeah.
Do you mind expanding on that a little bit, and we'll get the panel reaction?
First, I want to thank the panel. I thought each of the talks were particularly interesting today, but Dr. de Grey's talk I have some response to, and while there very well may be a stochastic aging phenotype, I don't think that phenotype extends to the neurodegenerative disorders, which is my area of expertise, because what a lot of people don't appreciate is that the pathology in Alzheimer's is not randomly distributed in space or in time, but is essentially invading the brain transsynaptically and retrogradely from what appears to be a portal of origin of the first cranial nerve. And Lewy body dementia, on the other hand, has been associated with the same sort of propagation from the 10th cranial nerve. And so this would explain the stereotyped longitudinal natural history of these diseases, each reflecting the brain revealing itself region by region to an invading pathological process. And the most likely explanation for that would be a infectious agent. And humans, as a unique species, are challenged by a whole family of neurotropic viruses, the herpes viruses, which are human-specific pathogens. And what this would explain neatly is why these pathologies tend to appear in humans uniquely and not in old animals in zoos or non-human mammals in old age.
Chuck, can I go first?
We took the whole conversation forward to whether you should actually kiss your grandmother. but we can step back from that basic level, and maybe, Aubrey, what do you think?
Well, sure. Okay, so first of all, I want to, let me actually first of all give a shout out to another researcher who's not here, but who has propounded this sort of idea very vocally over a long period of time and has been shamefully unsuccessful in getting adequate work funded. Her name is Ruth Itzhaki from Manchester, and her work on the causative effect of herpes simplex virus in Alzheimer's disease is far too neglected, I feel. But really, my main answer to what you're saying is that this, rather like the programmed, non-programmed aging debate, doesn't really alter our options in terms of therapies. The most, I think, unequivocal example in aging of something that certainly is caused by an infectious agent, but nevertheless is a progressive, cumulative problem that eventually causes ill health of a wider nature, and therefore we need to treat, is the effect on the immune system caused by viruses such as cytomegalovirus, which the immune system is unable completely to eliminate from the body, and which reactivate and eventually cause their cognate T cells to become unresponsive to antigen and yet to accumulate and fill up the immunological space, impairing the proliferative ability of other cells. This is something which, in my way of looking at things, is still absolutely damage accumulation and can be addressed by the same kind of approaches, such as, for example, eliminating these cells by pheresis, if one can identify them suitably selectively. The actual origin of the problem, namely the initial infection by CMV, doesn't actually matter. And from what you said about your hypothesis, I think the same applies, that even though there may be a very focal and perhaps exogenous trigger for the whole process, the actual effector things, the things that really do the damage, are downstream and can be addressed directly with just as good therapeutic consequences.
Well, it might very well be turtles all the way down, So, but I think where it would change your thinking is that you'd emphasize development of a vaccine, and then you would have a vaccine against Alzheimer's like we currently treat breast cancer and cervical cancer, instead of with genetic manipulations.
Yeah, let me say something about that, actually, because a lot of what I consider to be poorly thought out commentary has happened over the past year or so since the phase 3 clinical trials of anti-amyloid vaccines were reported. Essentially, everyone, or lots and lots of people, have been saying, oh dear, well, that didn't work, we didn't get any cognitive benefit, therefore amyloid doesn't matter in Alzheimer's, therefore let's focus on other things. I think that's nonsense. I think that that the result in those trials was completely expected, simply because whether or not it matters in Alzheimer's, it's certainly not the only thing that matters, amyloid. Alzheimer's is a multifaceted disease, and expecting the elimination of one feature of Alzheimer's to have a significant cognitive benefit is rather like taking a car apart and lining up all the pieces in a row on the ground and then pouring burning petrol over each of them and expecting them to move.
Right, but we'd have to then consider, though, the possibility that amyloid is actually an adaptation against a neurotropic human pathogen, and that interfering with it predisposes us to worse disease, or worse alternatives like encephalitis. So for example,
like a good offline discussion.
If you like.
In the original vaccine trials against amyloid, they appear to have succeeded in the sense that several of the patients have come to autopsy, but and there's no amyloid in there anymore. But regardless, they all became demented. All of them have extensive tangle pathology. And in fact, each of them who's come to autopsy, to my knowledge so far, has a Brock stage of 6 out of 6, which is the worst stage of tangle formation. And I worked with the Honolulu Asia Aging Study. 89% of the brains in that cohort have tangles in the hippocampus, but only 3% are demented. And among those are only 3 individuals who have a Brock stage of 6. So the way I look at that study is that eliminating amyloid from the brain accelerated tangle formation and led to extensive dementing illness.
So Don, let me thank you very much as a great bridge to open up debate and discussion. Did you have a question?
Sure. I'd like to focus on the relevance of the question of programmed aging for all of us here, which is whether signaling the body to do repairs is going to be an effective anti-aging mechanism, treatment, or whether the body's already doing its best to repair and we need to do the repair for it. So that's the question I'd like to ask each of you. Do you think that signaling the body to better do repairs is a global approach we should embrace, or do we really need to do the repairs for the body because it's already trying to repair it and not succeeding?
Who'd like to go first?
I'm not sure it's an either-or question. I think you have to take advantage of whatever strategies avail themselves. I mean, if we can develop small molecules that mimic some of the things that the body does in and of itself, and you can stimulate repair from within, that's great. In other cases, I think you'll have to go in with constructs or with cells or with new tissues. But I don't think it's an either or. I think it's a continuum, you know, of two, and quite a very big, broad continuum. And you just use whatever approaches make sense.
Yeah, I mean, I'd say certainly it's a continuum. The only thing is, at this point, we don't know which of those two types of therapy is going to end up having to predominate. We don't know how much we're going to achieve with signaling. If it turns out to be very little, then obviously we need to put that much more effort into augmenting the body's abilities.
So I see folks making their way to microphones, which is a great way of asking your question. Please. And do say who you are. And
Jay Jerome from Vanderbilt University. So to follow up on that same theme that's been going on and to state it in a way that makes sense to me. When we talk about diseases of aging, you're really looking at a very multifactorial situation, a lot of things coming together. Maybe Alzheimer's is caused by a virus, but why doesn't my 12-year-old daughter have it? And my grandmother does. There's got to be something else about the aging process. Perhaps it's the inability to fight off an infection. So I think what people have been saying here, and I just wanted to state it very clearly from my own mind, is that really we need to be looking at a multifactorial approach. Yes, if we can identify a causative agent, let's try to get to that causative agent, but that's going to happen when you're young, not when you're showing the clinical symptoms. And also, rejuvenation. Let's find a way to rejuvenate the processes that are going to help us deal with the fact that environment is there. We live. We got to live. We got to face that fact, and we're going to get damaged. How do we increase our ability to deal with that damage and to rejuvenate it and get back to the younger state? So you've always got to be a two-prong attack. It can never be an either or. You've always got to be looking at both of those. Is that reasonably what you guys are saying? I guess since I'm supposed to ask a question rather than preach.
Yep, it is.
But let me stir that a little bit, which is, you know, if it's multifactorial, if it starts at a quite young age, right, what do you do about it? How could any medicines be developed for that?
Go on, Tucker Bing.
Well, I think that there's in current preventive medicine, there's a good understanding that there are long-term processes. One example is in osteoporosis, and you can intervene long before there's a clinical issue. So, and even a conversation with cardiologist friends yesterday who have a hyperlipidemic family, they're giving statins to the children who are ages 7 and 8. So I think that's the template for the future.
Yeah. So the paradigm's there.
Yes.
As long as you can demonstrate that longitudinal impact.
Yes.
Yeah, of course, the thing to emphasize is that, again, we need this spectrum of approaches because those of us who are unfortunate enough not to be 7 or 8 anymore need something that is more actively rejuvenative, whereas those who are younger can perhaps benefit more from things that are more in the way of slowing things down.
Maybe it's possible to develop a biomarker for aging and then perhaps use that as a tool to then explore therapeutic approaches that can alter the presence of that biomarker.
So the biomarker that George Church was bringing up to attention was DNA methylation. How would that be considered as a biomarker for a targeted intervention? George here?
Yeah, I think stepped out.
Certainly measurable. Certainly measurable.
Yeah.
The concern that I have about that type of marker, and in fact about many types of marker, is that they may not actually tell us anything about what we might call biological age, about proximity to age-related ill health. For example, the paper that George highlighted in his talk actually showed this ridiculously high, like 96% R-squared of correlation with this particular set of methylation markers, but it was with chronological age, right? So it seemed to be completely independent of health, right? So, I mean, what the hell? Yeah.
I'm going to move us along a bit. We could talk about that for some time. I think the next questioner was on that side.
Actually, it was there.
I'll take the next one. Thanks.
So gentlemanly.
Yeah, thank you.
Ladies first.
I'm Jean Loring. I'm from the Scripps Research Institute. I'm a stem cell researcher. And the work that I'm working on right now, or one of the projects I'm working on right now, is a cell replacement therapy for Parkinson's disease. And it uses pluripotent stem cells, which are derived from the patients. So in a way, this is really just following in the footsteps of the work that's already been brought up using fetal tissue to essentially cure Parkinson's disease in some patients. So my question is really not about the viability of those therapies, but the fact that, let me give you the example. So Robin Williams was revealed to have Parkinson's disease just last week. And because of that, there was a press release from a company in San Diego saying that they had a therapy for Parkinson's disease available right now using the stromal vascular fraction from your adipose tissue. And essentially, they would isolate these, what are adipose mesenchymal stem cells, and re-inject them into you for a price, which is, they say is a clinical trial. The price is $15,000. And this has spurred an enormous interest among even the patients that I'm working with. Because this is actually considered to be a clinical trial, they really want to rush out and try it without very much understanding about the difference between a stem cell therapy and a stem cell therapy. So if you would discuss perhaps the great burgeoning of mesenchymal stem cells, fat-derived stem cells, for treatment for virtually all diseases, including diseases of aging, I'd be really curious in knowing what your impression is and whether there's any value in that at all.
Stephen, I think you should go.
No. No, the proof's in the clinical trial, and the idea that patients are paying to be enrolled in clinical trials, I think, is unethical. Clinical trials is this safe in the first instance, and secondly, is it efficacious? And you have to prove that. To say we have a clinical trial and we're going to do this and you're going to pay for it, that's a treatment. I think companies that have these kinds of approaches are unethical. That's not to say, and on top of it, I'd question the biology. How would MSCs derived from fat impact on Parkinson's disease? Show me, proof of concept. I think this is just, snake oil.
I sort of figured you'd think that, but how about the rest of you?
Let's keep it going, please.
Okay, my name is Ben Schwergold, retired from Dartmouth and currently on my own. So first of all, I agree with everything that's been said, nothing too controversial. And I just want to give my overall review and then a question. So first of all, until 1800, as was shown before, lifespan was about 35, 40 years. And since then, it's increased. With its increase, we have become aware of more and more diseases and factors which influence lifespan. And the most effective measures have been low-tech measures: sanitation, vitamins, better nutrition. And it is my firm belief, and I take some issue with Dr. Finch, is that some of the most effective interventions with aging, not for everybody, will be low-tech, what I would call second-generation vitamins, which have been neglected. But if you look at the record of iodine, vitamin A in milk, et cetera, these have been proven extremely effective, and clean water, et cetera. So I think the future will be twofold or multifold. There will be public health measures and cheap interventions, nutritional interventions, along with the high-tech stuff. So I think the dichotomy between the developed and undeveloped world will probably not be as great as you fear. Because
Great.
as
I
we
think
find
we
out
get it.
about these nutraceuticals and supplements, which for some reason have not been researched over the last 50 years, we will find that there are things we can still do to prevent obesity, heart disease, diabetes, etc.
Well, I certainly agree that there are a lot of low-tech, easily treated things. For example, vitamin E deficiency is quite common, and that's a cheap pill. But the issue is to, on an educational level, both of the population as well as the local healthcare deliverers, and that's a long-term global process, as we can see with the response to Ebola in West Africa.
Okay, just one comment and I'll shut up. What I bemoaned is the fact that investigation into nutritional components and supplements have sort of fallen off with the burgeoning of molecular biology, DNA, stem cell, high-tech stuff, and I think there's still a lot of stuff to be discovered.
Absolutely agree.
Hey there, I'm Shane from Stanford Med School. Main question is for Jeff. It's kind of a two-part question. First, could you elaborate on why the stem cell niche seems to be depleted with age and why the stem cell population is depleted? And kind of second part is, my limited understanding is that it's driven a lot by senescence. Partly that's thought to be a good thing in that it's, these stem cells might go on to become cancerous, so it's a preventatory measure. But is it also possible that the senescence is driven by inflammation and that undamaged cells might become senescent and that therefore we could target the senescence pathway perhaps for intervention and reverse that and then get some sort of increase in rejuvenation and increase in stem cell population?
Wow, that's a great question. I think there was more than two. So
yeah, sorry about that.
And I really would say I don't have that much. I think there's no direct answer to probably any of the questions that you've asked. I think that there's some sort of nuances in terms of as people age and you have more degeneration happening, then you have more demand for the stem cells that are still present at that time to work in overdrive to then fuel that. And then as you mentioned, there's this senescence that's happening. And I would turn to that. I'm sure there's probably people in the audience who could better answer your questions than me. So I think if anybody wants to give a go at any one of those.
Okay.
I see no hands going up, actually.
Yeah. But I mean, these are
Really
these are
questions, yeah.
yeah.
Thank you. Martin Borg Jensen. I'm at the Buck Institute. So the Bentley is still running because we can just replace all the parts. But in the Bentley, the brakes aren't telling the cylinders how to do their job.
Yeah.
So in a human, that would be the case. And that's obviously very complex, which is why we haven't fixed aging yet. And we're working on it in basic science, but it seems to be extremely complex. Do you think, to the panel as a whole, do you think that there is a solution for fixing things without understanding them, given that we have a very interconnected biological system? And do you think that we could and or should try to get some kind of standardized measure of whole body effect on aging when we do individual treatments?
All right, I'll try. I'll have a go at that first. So first of all, I think that absolutely the whole damage repair paradigm is motivated by the concept that it is a very powerful way to sidestep our ignorance. Essentially, we can divide the process of, goes from being alive to being dead into these two parts. The part that happens lifelong, which is the accumulation of molecular and cellular changes to the structure and composition of the body, and the part that happens late in life when those changes progress to a point where the body is not set up to tolerate them properly and function starts to decline. And the wonderful thing about going in at the level of repair of damage is that it uncouples those two processes from each other. It means that damage can still be created. We don't have to worry about that, and therefore we don't have to understand how it's happening, really. And the damage, the link between having too much damage and having pathology is also something we don't need to care about and understand very well anymore, because we're not letting the damage get to the pathogenic threshold. So that's the whole principle of the thing. In terms of whether if you said whole body monitoring, so to speak, of biological age could be done in the context of these therapies, well, of course, we've already had the question about whether biomarkers of aging are possible and the difficulties of finding them. I would say that we shouldn't get too hung up on that. If we start off by developing individual therapies that are targeted to particular aspects of aging, then we can always, first of all, develop and test them in the context of people for whom that aspect of aging is premature, for example, and so in relative isolation, and then we can incrementally start to combine and get to a point where we are truly comprehensive.
Guillermo Garcia Cardena from Brigham and Women's Hospital and Harvard Medical School in Boston.
Sorry, can you speak up a little bit?
Yes.
Or get closer to the microphone.
I just want to follow up on some of Audrey's comments and also follow it on the last question. I think that one critical thing that we need to define and start thinking from the point of view of the research is to make a clear distinction and define targets between lifespan and aging as a process that Audrey was referring as a whole cumulative effects of accumulation of defects. And one of the critical things, what we have learned in the last few years is that there's things that can regulate lifespan, and there could be some clocks in the cells, and we can think about telomerase and telomeres as some of the cellular basis of that. But also, there have been very few pathways that have been defined to be basically really driving the aging process. And I can think about two particular, one of them is the clotho gene, in which basically you have either gain of function or loss of function, but you can recapitulate everything that happened in the aging process, not only in the cardiovascular system, but also in bones or in the skin or in some parts of the brain. So I think that it's going to be critical as we move in the conference is to try to think about that. So diseases of aging as such that are like the consequences of the process, and some really fundamental pathways that are making the physiological system to go through the process of aging. So I just wanted to contribute on that.
Thanks for that. Since we have just two other questions, why don't we have those two questions and then we can ask the panel to respond to all three.
Okay. Daniel Roth, Common Gene. Question is to each of the panel members. What do you think it takes to dramatically increase the amount and quality research in aging in academia?
Money.
Money.
Okay, and then...
You each believe that that's what it takes?
Yeah, I mean, in particular, if you look at the funding structures now, if you're not doing translational research, your chances of getting funded, particularly in Europe, are nil. And I think, we're going to miss out on a lot of fundamental biological insight that, will just be lost, because if you can't tie it to a disease or tie it to pathology or tie it to therapy, you're not going to get funded. So I think that's a gross oversight of the funding agencies, this drive toward commercialization and everything having to be translational.
So I'd like to come back to the earlier question, aging versus lifespan. But sorry, there's just be one more question from
So Richard, you at the beginning expressed that it seems disappointing that not as much has come out of this sort of field as you might have expected 25 years ago. And I think that in the light of the focus on translational research, we might expect a slowdown in the long term, but we might expect an acceleration in the short term. So that seems to amplify a mystery about why so little might seem to be happening. And I really want to hear your opinions about that. And for the panel as a whole, if we adopt a damage perspective on aging, does it seem like the simplest organisms, the ones with the fewest tissue types, the best understood tissues, such as nematodes or C. elegans, might be a more promising place to look than in humans?
So if I may, the first question of those three, comments on that point.
You're going to have to remind us what it was. It was your fault that we got three questions in a row.
So it's entirely I could always just answer that last one. No, so this was the point about aging versus lifespan. No.
Well, yeah, okay. So we've seen maybe 15 or more years of increase in life expectancy in the industrialized world since World War II. And the great news is that pretty much all, depending on how you measure it precisely, pretty much all of that increase has been an increase in the time that we stay healthy. In fact, Dave, your average 70-year-old today in the Western world is about as healthy as your average 55-year-old was in 1950. This is fantastic news. And so in a sense, we can say that the kind of rhetoric that often gets bandied about in this field that tries to distinguish aging from lifespan and talks about things like, oh, it would be good to compress morbidity, this may actually not be the right way to be thinking. If we can simply postpone morbidity at an accelerating rate, eventually we'll be postponing it 4 times faster than we are at the moment, and we won't ever get to it. You know, that's the goal.
I was just going to answer the thing about the funding, but
okay, so let's move to the funding because we didn't quite finish that discussion.
Yeah, no. Just one point is that I think securing grants in many ways is kind of a dance, right? And it's like you need to be in rhythm with your partner. And so the partner in the dance are the funding agencies. And in many ways, I think you have to learn the rhythm of the agency and then tailor the grant to maximize impact or success. So I think it's one of these, there potentially is challenges in getting funding for certain areas, but if you can tool the grant to better understand what the funding agencies are looking for, you could potentially fund your research, but not necessarily write it the way you would have without knowing what the grant agencies are looking for.
And of course, this is exactly the opposite of what Shannon and other founders of the NIH intended with investigator-initiated research projects. Now it's heavily dependent upon the local culture in each of the branches of the NIH, across the NIH, and the program officers who, in unquestionable high intentions, are nonetheless acting as independent policymakers on what is relevant at that time in their shop.
So it sounds like the basis of a great satellite masterclass that the two of you can run on how to get grants out of agencies, how to do this dance. Because I think there's a fundamental point under what Steve said, which is what these agencies choose to regard as the big problems and how to go about it has an enormous effect on the distribution of money.
It does now.
And actually, I can just slip in that a little point about the central mystery that I mentioned. I'm not at all sure that things that are labeled translational research actually are translation. But,
well, it's as Jeff alluded to, it's part of how you play the dance. When all of a sudden there was huge amounts of money in the UK for stem cell research, people who didn't know a stem cell from several other things, all of a sudden we're doing stem cell research.
What do you know?
Exactly. So
And we'll leave whether we should be studying worms rather than us, maybe for another day, because I see Jerry has a question, which may be, is anybody hungry?
That was exactly my question. Is anybody hungry?
Yes.
All right. Lunches, just go out the back doors and around the corner, and lunch is served. So.
But first, may I thank my colleagues on the panel.